Գիտության և գործարարության օրեր 2022 Հայաստան։
Ladies and gentlemen, it is with great pleasure we are opening our final session to the day. I'm your host, Professor Armen Kherlopian. We have with us Armenian-American scientist, Dr. Ardem Patapoutian. Please join me in a rousing welcome. In terms of the structure of our time today, we'll be beginning with a bio sketch, followed by Q&A, followed by open Q&A, and subsequently some closing remarks. So with that, and without further ado, Dr. Patapoutian is a molecular biologist who's conducted seminal work in the area of sensory perception and transduction. With his team, his contributions to science have included the discovery of novel ion channels, which are behind the sense of touch, sense of temperature, and even proprioception, our awareness and sense of space. Dr. Patapoutian attended the American University of Beirut before he immigrated to the United States in 1986. He graduated from UCLA in 1990 and received his PhD at Caltech in the lab of Dr. Barbara Wald in 1996. After postdoctoral work with Dr. Lou Reichard at UCSF, he joined the faculty at Scripps Research, where he is currently a professor in the Department of Neuroscience. He also held a position at the Genomics Institute of Novartis Research from 2000 to 2014, and Dr. Patapoutian was awarded the Young Investigator Award from the Society of Neuroscience in 2006 and named an investigator of the Howard Hughes Medical Institute in 2014. He is a fellow of the American Association for the Advancements of Science and a member of the National Academy of Sciences. As a biophysicist with interest in research associated with machine perception, I can say on a personal level, it is an absolute honor to be here with you, world-renowned expert in sensory perception. So with that, please welcome again Dr. Patapoutian, Nobel Prize Laureate. Thank you for being with us here today, Dr. Patapoutian.
Pleasure to be here. Having a great time here in Armenia.
Absolutely, and let's kick off. How did you begin in science?
So I grew up in Lebanon, so I lived there in Beirut the first 18 years of my life. And so when I immigrated to the United States, my parents had decided that I should go to medical school. I say in jest, but that's really how it was, because my parents really wanted one of our kids to be a doctor, and both my sister Huri and brother Ara, who were there, were really very squeamish. Like, there were lots of large cockroaches in Lebanon, and whenever we saw one, they were scared to kill it, and it was my job to go and get rid of the cockroaches. So I was thought to be the person that I should become a medical doctor. So when I went to UCLA in California as a pre-med, it was a big state school, so class sizes are 200 to 300, and I knew to get into medical school I needed good letters of recommendation for medical school application. Unfortunately, it was very hard to meet the professors in person so they could write good letters. And I decided that I should work in a laboratory to get to know them well enough so they could actually write me a letter. So for completely devious reasons, I started working in a laboratory. But within weeks, I absolutely fell in love with the discovery process. And as they say, the rest is history, because within a few months, I changed my mind and decided that doing research, getting a PhD was more of a better fit for me. and I've been doing research ever since.
Well, I have no doubt your original drive to do research just for a letter is now coming back, and many people are now requesting you for letters of recommendation. You mentioned a bit your family. So I had the pleasure of meeting your brother, Ardem Patapoutian, who himself has a PhD in electrical engineering and over 120 patents. What was it like growing up in your household? Yes, round of applause.
You know, my parents, Sarkis and Haikuhi, were very dedicated to education. You know, they have that Armenian knowledge and spirit that we come from a disadvantaged background, and the way we get ahead of in life is to first and foremost have a great education. And so I remember, especially my mom, we were not the best of students perhaps because we were not very motivated, and she sat next to us and made sure we did our homeworks and took a lot of care, going back and forth between me and my brother to make sure that we did our work. My dad was more on the side of what we call tiger parent, I guess, in United States, where I used to come and tell him, "Hey, Dad, I got a 95 over 100," and he would say, "Why didn't you get 100?" And so that's my memory of my father encouraging me. But I guess one needs both to be challenged and to be nurtured, and I think I got that from both my parents, who insisted that we take education very seriously.
And in terms of that focus on education early on, what was it like in Lebanon? What organizations were formative in those early times, outside the family context as well?
So I think most people in maybe who live in Armenia think of us from the diaspora as very integrated, so Lebanese being Arabic our first language, but it wasn't the case. So we grew up Armenians first. Our first native language is Armenian. All the schooling and sports club, the first 16 years of my life were all actually centered around AGBU, Armenian General Benevolent Union, who had the Demirjan Elementary School, Hovagimian High School. So those are the organization that's played a huge role. And it was during the war, so there was, what you do outside of school is very important. And we had a wonderful, very advanced club, Alex Manugyan Club, where we, played sports and had cultural events. So these Armenian organizations, especially AGBU, played a massively important role in my childhood.
Indeed, there might be an analogy there in terms of some lasting institutions, the Nobel Foundation being on the back of invention, and then similarly for some Armenian organizations. You mentioned your parents briefly. Where are they from, your grandparents?
I think like many immigrant Lebanese stories, my grandparents were very young when they, had to leave Turkey and get to first Syria, then Lebanon. They've also spent some time in Cyprus. My father, for example, his family is from Hajin, and so anyways, the important thing was that they all remained Armenian first, although they were, came in and even born. They were born in Lebanon, but their parents came to Lebanon from Armenia, today's Turkey.
Thank you for that context, Artem. In terms of the Nobel Prize announcement, I understand also your father also had a role in that. I would love to hear how you heard the news.
So the Nobel Committee decides the final who's going to win the prize in the afternoon in Stockholm on October 5 last year, for example. And after they had decided, they called the people who are going to receive it. Unfortunately, that's about 2:30 a.m. in West Coast, California time, where I live. And I had my Do Not Disturb on my phone, so 3 phone calls from Stockholm were missed. And then they found somehow my father's phone number is another Patapoutian from California. So my father and mother, who are 95 and 90, still live in Los Angeles by themselves, I'm proud to say. And so my dad got a phone call from Stockholm, and the guy who is the head of the committee told me later that my dad has kind of yelled at him of why he's calling him 2 in the morning, and then proceeded to then call me and tell me that there's a call from Stockholm, I think it's important. And so I heard it through my father first, which was a very, very unique experience in a way. And as soon as I looked at my phone, there were multiple missed calls from Stockholm, so I kind of had a hint of what's going on.
Indeed inspiring to hear, and fantastic to know your priorities is that you will definitely take the phone call that matters the most. Wonderful. So as we transition here from the personal context, Dr. Patapoutian, would love for you to help us understand the nature of your discovery. And as a special treat, we have a select set of images from your Nobel Laureate speech. So we'll be bringing those up. But first, if you can get us going, and as you see here, we think of our various senses. For example, the sense of sight, where light impinges on the retina and then is subsequently processed by the lateral geniculate nucleus and primary visual cortex. So there's light being transduced. We have our sense of sound. Earlier today, for example, we had a colleague, David Baglasarian, talking about CRISP in terms of processing of that type of information. And then there's the chemical nature of our sense of smell. And would love to hear your overview on sensory perception and specifically the sense of touch.
You know, for the longest time, people, of course, have studied sensory systems, and this has taken us over 100 years. Well, our specialization is at the molecular level, in a way, the gateways of these senses, and that is how do you translate these stimuli from outside our bodies and how we sense it and do the interpretation in our brain. And so for visual system, as you said, it's visual cues, photons that are being sensed. For taste and smell, it's chemicals. For touch, it was just this enigmatic, kind of much less studied and understood of pressure, right? So when someone just touches your fingertips, just imagine it within milliseconds, something's happening under your skin that you're sensing this slight indentation of the skin to make you realize that someone's touching you. And that process at the molecular level of how it was known is not understood. So my laboratory found the molecular machines in a way that translates this physics into chemistry, sometimes I like to call it, because that's what you're translating pressure into a language that your brain can understand. So why is this important? I think just understanding the biology is fantastic. For centuries, scientists have been driven by curiosity, not necessarily by applications. But of course, as many important discoveries, this has important applications. For example, pain is a medical condition that's relevant to almost all diseases, and yet we don't have adequate pain medications for many types of neuropathic pain. And the research that we've done, some of it actually is already in clinical trials to see if manipulating these receptors, these gateway proteins in a way for understanding these mechanical forces can become future medicines for indications such as pain. But as we've learned over the years that this pressure sensing is relevant to much beyond the somatosensory system of touch and pain, but relevant for blood pressure regulation, for function of the urinary system, even for malaria it has some relevance. So that's one of the beauties of basic science. It's a luxury to just answer these general curiosity questions. But again and again, science has shown that discoveries that were done in that spirit later on, years, sometimes decades later, have found incredibly useful applications for humanity.
Yes. And before we go deeper, specifically on the molecular level, we have in the image here a rose. And so you made reference in terms of the scale or the gradation. So we can identify a rose by seeing it. We have cues in terms of its pleasant smell. What can you talk about specifically on the sense of touch and also the sense of pain? After all, a rose has thorns.
Yes, I've picked rose for that reason, because it has so many sensory aspects to it. There's the visual, there's the smell. Most people don't eat it, but you can use it in cuisine. But of course, the sense of touch is both the soft petals to the thorn that's causing the pain. And what we know is that you have specialized neurons that sense different aspects of what I call somatosensation. Some are very specialized to sense the light touch. Others are specialized, what we call high threshold receptors, where they only respond when the stimulus is damaging. And the idea is that this acute pain is absolutely essential for our survival, because it tells us what we should avoid and what we shouldn't. When pain becomes something we want to use medicines against is when it becomes chronic, which means the response, the painful sensation remains even when the initial stimulus that caused the pain is no longer there.
Well, it makes absolute sense that an Armenian scientist will make discoveries related to identifying and alleviating pain, as per the expression "Savat Danim." We'll return to some of these concepts subsequently after we finish the portion on discovery, specifically the rise of ventures that are using the sense of sight, like Skyla, Mano, Cognaze. We heard earlier about CRISP, so it's quite exciting, the implications for our economy, this interface between sensory perception and machine perception. And so, Ardem, before we transition to our next image, can you help orient us in terms of your engagement with science? Specifically, you made reference to the pivotal moment of needing a letter, but could you provide us some insight in terms of how that research activity, early research activity, captivated you? What sort of environment did you need that enabled you to be inspired to pursue such a career?
Absolutely. We talk a lot for Armenia and the whole world, the importance of education and how to teach education. So I feel like I stumbled into research, and I think part of the reason I had to stumble into it is because even in the United States, I don't think sciences are often taught in the best way possible. So I thought that I really disliked laboratory research or experimentation. The reason is laboratory classes, the way they're taught, is complete opposite to what happens in a research laboratory. So you go into biology lab usually, and you're given an experiment, you know what result you're going to get, and you go through difficulties to get that result. I think people often confuse scientists for people who have lots of knowledge in a certain amount of field. But I tell you that scientists are mainly ignorant, and they love the ignorance. And so when I go into lab, I don't talk about what we know, we talk about what we don't know. And that's what's so exciting to us. So once you identify what you don't know, and then the whole idea is to come up with experiments and ways to try to find out how something works. And that is such a delicious thing to do. I can't explain it in words, but this is what's captivated me, is to explore what we're ignorant about, and then come up with ways to be less ignorant. And that process is what's really captivating. And then the other thing I would add is, when you're doing research in biological sciences, you have those rare days where you look at a microscope or a gel in molecular biology, and you realize that you just understood something about how our body works that nobody ever knew before that. And again, it's very hard to describe the feeling by words, but that's what drives most scientists that I know, is that feeling that you discovered something that nobody ever knew before. So those are the things that got me really excited into life science research, and that's what I've been doing these decades.
In the Armenian language, the word for research, hedozodel, translates as research. It's also very closely related to explore.
Wonderful.
And so we have this concept, and then we also have an idiom, chigarali, which means not possible, to say maybe you shouldn't try that. How do you think of this dichotomy, and what needs to be emphasized in terms of being able to do just that? To explore, to do research, to be comfortable with the unknown, and subsequently make discoveries.
That's one of the most important, I think, aspects of doing research, but I would think for almost any careers is, what are your goals? How achievable are they? Are they impossible? And there's lots of subjective thinking, I guess, because some things might be impossible within the next few years. And so what you invest your time in, all your work, of question of how important it is difficult to teach. There's lots of instinct involved, which of course comes with experience. And I think, the famous saying that luck occurs to the prepared mind kind of plays a role in this, because you need to have done this enough to see what works, what doesn't, and get a intuitive feeling of what is a good thing to invest in. It's difficult to explain exactly, but I think it's something that you need to have resilience, you need to be persistent, but you need to ask the right questions.
Let's proceed along as we were discussing about the key senses we have. We often think about them as 5 core senses, but there are others. So we have another image here from your Nobel Laureate lecture. Can you talk to us about our senses more broadly? What other senses do we have? What senses might not actually be a sense, like a sense of direction? If you could orient us how to think about this.
Absolutely. I think one should first of all realize that humans are not the only species on this planet, and there are lots of animals that have echolocation that we don't. Some have infrared sensing. So, we live in a world of physics and chemistry, and each species' senses have evolved to make sense of their environment. And for us, life is about these 5 senses, but almost philosophical, but to realize that you're sampling a very small portion of the world we live in, and that's what we've evolved to sense. So that's our sense of reality, which very strongly depends on the specific types of senses that we have. So coming back to us, yes, we call the 5 famous senses, but especially for touch, which sometimes I call somatosensation, is part of a much larger sense. So clearly we sense temperature, right? Cold and warm. It's not represented in one of the 5, but I kind of put them under the somatosensation circle. Even just the touch, I think this slide highlights that, of course, we can discriminate touch, but the benefits of psychologically a hug from a loved one or a caress of a mother, these are, the very different information you're getting from touch than something's touching my arm, I need to move it, right? And in the brain, these actually are connected to completely different centers, so that one is giving you what we call affective touch, giving you emotional satisfaction. The others are just telling you where things are. So that's one aspect of it. We talked about pain. Is pain part of touch? Is proprioception, sometimes called the 6th sense? For those who don't know, proprioception is your sense of, most literally, it's where your limbs are in space. So if you all do this exercise of close your eyes, extend your fingers, Now I have a very good idea of where my hands are in space, and I can bring them in and touch my nose. So the reason I can do that is because I have sensors within my muscles, and from how much each muscle is stretched, over the years when I was growing up, I've started projecting this image in my head of where my body lies. That's why babies are not very coordinated. They don't have a well-developed proprioceptive system yet. But we do, and we use it very effectively to just sit down, to stand up, to walk, to dance, all these things that you take for granted. But without proprioception, as we know from some of the clinical studies we've collaborated with, is all those things that I mentioned you can't do. If all of a sudden you lost your proprioception and you tried to stand up, I would fall down because I would have no control over my body. Sometimes I say this is the most important sense that you don't even know you have it, which is pretty interesting.
And so if we take the example, and you have a stunning photo here of Yo-Yo Ma playing the cello, what is going on? And in particular, how is a link possible to be made from the molecular level to something as beautiful as playing music?
Well, that's a lot we don't know in between those two still. I mean, this is why the receptors are important to find. I compare it to, in a scientific question, that there's a room and it's very dark and you don't know what works. And how are you going to study it? The first thing you have to do is find a key to the room, open it, and then start studying what's inside. So these receptors, in a way, are those keys that opens the room, and now we can start looking, what are the consequences of activating these receptors? Are my receptors different than yours? How does experience affect how these receptors are used? Is someone like Yo-Yo Ma who makes beautiful music, is his proprioception enhanced compared to others? Is Michael Jordan have much better proprioception than the rest of us? Those are questions that we still don't understand, but we can slowly start answering them.
And so while we're on the topic here in terms of the receptors, can you provide some intuition in terms of order of scale? We had colleague Dr. Garig Israelian talking about black holes, so very different time and spatial scale earlier today. And we have here a visualization associated with the landmark discovery of the ion channel associated with the sense of touch, piezo to be specific. Can you help orient the listeners in terms of what an ion channel is? Why the sense of touch on a molecular level has been so elusive, please?
Well, the reason it's been very elusive is these are, first of all, very, very nanoscale level machinery. So imagine that your neurons that sense touch and proprioception reside just dorsal to your spinal cord. And they're these tiny neurons, very heterogeneous, and then they send this thin process all the way to your skin and muscles. And at the tips of these neuronal projections are these little sensor proteins that get activated when you move or sense something. And so there hasn't been an easy way to isolate them. You made the analogy to the vision. So the eye is in one place, and people have been able to take material, isolate the receptors from it. These are distributed all over the body in very small quantities, so it's been very difficult to identify them. And so now if you bring it back and say, these are nanometer wide, tiny proteins that even light microscopes can't detect them. So your evolution, how would you design a sensor that would be able to sense pressure and translate it to an electrical signal, which is what they do? And what evolution has come up with is this relatively very large and spread protein in the periphery of the cell. So all of your cells is covered by this fatty lipid, what we call a bilayer, to protect the cell from the outside. But if you have complete protection, if you're a complete isolationist, then you have no way to communicate with the outside world. So then evolution has to create these ways to allow communication to the outside cell. So it's designed this monstrous looking molecule that can go up and down a fatty layer 38 times and spreads around this fatty layer. And then when this fatty lipid gets stretched, the protein itself gets stretched, which causes it to open. Once it opens, it creates a hole in the cell where specific ions like calcium can flow in, and this is sufficient to start the signal that a few milliseconds later you register in your brain that something touched me in my fourth finger.
So in terms of these various scales, we can think about a meter, our perception here, the size of a person, 1 to 2 meters. Then we go to the millimeter scale, aspects of different organ systems and tissues. And then the cellular level overall, micrometers.
That's
and then
right.
you mentioned nanometers, so still 1,000 times
That's
smaller.
Right.
So how many ion channels would be on a cell? Or how can you help build our intuition for something we cannot see as plainly as the meter scale?
Yeah. We actually don't know the answer to that because they're so difficult to visualize, but we estimate that every cell has about 1,000 of these little piezos all over itself. Just to give you a scale, so a grain of rice is about 500 times bigger than a cell, so you will not be able to see it. And if you enlarge a cell to this level, each of these piezo proteins will be a dot that you won't even see. So we're talking about very, very small scale.
You'd also mentioned in terms of the movement of ions, specifically charge. So I want to discuss a little bit beyond the spatial scale, the temporal scale. So for example, homes in a city have on the wire associated sound check. All right, we're back. So in our homes, there is a negative charge, so electrons going down the wire, powering our appliances, our cities. In the cardiac system, for example, it's the movement of potassium, as well as sodium and somewhat calcium, which is positive. That's mediating the electrical activity. So can you speak about this type of activity in the sensors you've studied, and as well as the time scales we're talking about?
Sure. So most cells, there's a membrane potential difference at this fatty layer that I talked about. And when something like the piezo ion channels open, you get more both sodium and calcium. In most neurons, sodium is the one that drives the change in membrane potential. And when this happens, there's more downstream steps that pretty much depolarize, and a small current actually is transmitted across the neuron, and that's actually the language that one brain cell, a neuron, talks to another. So it's the same kind of ions involved in heart, but sometimes the direction is different. With respect to this time scale, it's all happening in millisecond and sub-millisecond levels. And so we don't even think about that there's any time delay between something touching you and you perceiving it. There is, but it's at millisecond levels. And these are one of the fastest signaling systems in our bodies. So for example, your sense of smell or even taste is considerably slower than your hearing and your touch, which are all mechanical based.
So here we have the protein coming together, mediating this electrical activity, and then operating on a spatial scale of nanometers, near instantaneous in terms of time scale. Our understanding that this is the largest such receptor discovered. What is the current thinking in terms of why there's so many transmembrane domains?
We don't know. We think it kind of plays an amplifier role. It's kind of like, think of an octopus spreading its arm across the cell, and so it has sensors, antenna at many different directions where it can summarize all the mechanical distentation that the cell is experiencing, then to initiate the signal. This is pretty much all we know about it. again, evolution is a very useful tool for biological studies, and piezos have relatives not just in plants, animals, but even unicellular organisms. And in every one of those, the size is still very large. And if evolution has spent the energy to keep making this in a large scale, you know that there is good reason for it, because evolution is very stingy. If something's not needed, it would cut it out. So these receptors have remained large. And I know it can be confusing because I keep talking about how small they are and also how large they are. It's relative. All proteins are small, but compared to other ion channels, these are large, just to clarify that. But that's why we think the size is important.
And we're going to shortly jump to the human scale. One question here, Artem. So you made reference to evolutionary preservation. Are you saying that the very same receptors that are so the same receptors for the human touch responsible for how a plant or a tree would find water?
Yes, I think this kind of biological evidence should give us humans some humility about our part that we play in this world, that the same exact type of molecule is used by plants to detect how tough the surface, the soil is that they penetrate. The same exact family member of molecule is in our fingertips and helps us sense touch and pain. I find that one of the most fascinating things that my lab was able to identify. And again, it just shows that also evolution, once you find a problem solving from a question that from a problem that you have a nice answer to, you use it again and again and again. And again, this is useful for evolution and many other aspects of processes too.
Indeed, this is tremendous. The implications of this discovery from your group, colleagues over the years, having so many far implications from the human sense of touch, potentially leading us to pain management strategies, to even implications in agriculture and beyond. When we were looking at the previous image from your Nobel talk, there was also a cat with the effective touch title. And I want to bring to bear one of the images appreciated most in that lecture, but also one of the images appreciated most by the Office of the United States Presidency. Specifically, when Security Advisor Carlton Philadelphia brought his family in what is known as a departure visit. After a set of years in public service, the president wishes the family, the public servant well. And here we have something quite striking. So Carlton's son, Jacob, is touching President Obama's hair. Could you elaborate or provide some context in terms of what's happening on the human level?
Absolutely. So I think the main point of this is that we don't just use the sense of touch to sense texture, but I think this is such a powerful image for a young African American boy to realize that after all the atrocities against that race in America, that there is a guy right next to him who's the President of the United States, and the hair feels exactly like his. And so I think that's one of my favorite images that illustrate the power of the sense of touch, and also the power of representation and just identifying yourself with someone and giving hope that if Obama can do it, then this guy can do anything he wants.
Indeed inspiring. And I believe in recent news and current affairs, President Obama has now spoken with the boy again, but on the eve of his high school looking toward college. And so this power of representation, it is indeed a pleasure and privilege you are here, Artem. And as I serve on the scientific advisory board of one of NASA's institutes, I can imagine your schedule this week is that of like a NASA astronaut on the space station. So we had the pleasure of welcoming you at the American University of Armenia commencement speech addressing our students. Would love to hear what you've been doing, how you've been engaging, and if you've been having any sleep at all.
As you all know, if you travel 11 time zones, you have very difficulty sleeping. And so that on top of the kind of schedule you're talking about has taken a toll, but I'm not complaining at all. I've had one of the most amazing few days to see all the people who want to take selfies with me or just want to say hi, and the great reception I've gotten from scientists to government officials here is really special. I think that scientists usually are hidden in their laboratory doing work, not really have great social skills, and to be treated like this is a huge privilege, and it's really testament of what Armenia recognizes as important, and I'm so proud to represent that. There was even a stamp with my face on it. I know lots of other Nobel laureates back in the US, and I make sure I tell them all, and I can say that everyone is so jealous about the stamp. No one else that I know has one that's alive, so in science field, so that's a really cool thing. But really, I think I really honestly don't think of myself as a role model, but I'm starting to realize that it might have that effect on some young students who are interested in science and technology, and if my example is used as if Ardem can do it, then I can do it, then I think that might be my biggest contribution, and I'm very happy about that.
I'm so delighted to hear about the fondness of the stamp. And indeed, Ardem, you are in good company. I believe the immediately preceding stamp was for scientist Alexander Khomudjan, who designed the first rover that walked on the moon. So delighted to see this chain from biotech to space tech.
Nice.
You made reference to your team, and indeed science is a team sport. And so would love for you to take us through in terms of how you think about leadership, science, starting from your team and then expanding outward from what you've learned.
Yes, I think this is so important to emphasize, so thank you for asking. Prizes are given to individuals, and our society celebrates individual accomplishments, and of course I wish many more Nobel Prizes to people of Armenian origin, but also it's very important to emphasize that the work that I was recognized for is really the benefit of so many other people. So there's about 16 to 20 people in my laboratory at a time, and we all work together to come up with ideas, and they do the experiments, we do the interpretation. So over the years I've had over 80 people work with me, and then we also not working in a vacuum, we work in other fields. Our discoveries were built on discoveries of other scientists who've done it, and so it's just a very important thing to emphasize this group effort, this working together in a group and having joint goals and aspirations. And it becomes difficult because after all that, the award comes to me, for example, but it's very important to share the credit.
And with that, we have a snapshot of your team here. So not quite 80 people, but a moment in time in 2018, I believe. So Artem, one thing that is incredibly striking and inspiring about this photo, beyond everyone seeming to have a great time, is how international the team is. How are you able to recruit, inspire, engage? And on one hand, 80 sounds like a large number as far as teams go, like over time, but it also sounds like a tiny number given the impact and potential implications. So how do you think about this, the scale of high performance teams? Would really appreciate your thoughts.
Earlier you asked me about also what I loved about science, and this international culture is something that really attracted me from the beginning. You know, when I started working in the lab as an undergraduate, there was someone from Spain, there was someone from Iceland, there was someone from India, there was, you know, all over the place. And this kind of diversity really contributes to creativity because everybody comes with their own history, their own knowledge, their own, you know, understanding of how they look at a problem. And so now we often talk about diversity as something, you know, we should do to be fair, which I think is great, but I also think that there's inherent value in diversity. And this international cooperation, I think, was most evident during COVID. As you know, the world was hit by this pandemic, and if the various governments and societies can learn from scientists of how they work together to, within a few weeks, first of all, identifying the virus, knowing its sequence, and then in record time providing vaccines that kind of was able to get us out of the pandemic much earlier and with much less death and suffering than would have otherwise. I think it's one of the greatest achievements of science. And if you look at how all of this happened, such a huge international effort, including, of course, Mr. Afeyan Nubar for Moderna and the vaccines that the company that he started, the fellow Armenian. And so anyways, I think international cooperation is not only enriching on a day-to-day basis, it's a very powerful way to solve problems. And again, I think science has lots of things to teach society, and this kind of cooperation is one of them.
Indeed, the pandemic is particularly trying and is in many ways still ongoing. And in a recent peer-to-peer interview with Nubar Afeyan, so the critical work at Flagship Pioneering regarding an innovation and scientific process, and subsequently his leadership as chairman of Moderna, making a difference for the world, begins to bring the question, we have titans such as Nubar and yourself. This past year, we've had health tech robotics companies, for example, Embodied being featured in Time magazine. Another company also, Robin the Robot by Expert. We have algorithmic capability in terms of bioinformatics under the umbrella of bioscience and, you know, adjacently biotech. What else is needed? We have the inspiration, we have the proof points of small teams. How would you think about this?
I heard all your statements. I didn't hear the question. I'm sorry.
The question is, we have the inspiration. What else is needed to sustainably build up these industries in Armenia? That we transition from striking and impactful examples for the world to strengthening the economy of the country.
I'm quite optimistic. I think if you look at the high-tech industry already, there is so much going on. There's so many great companies now, some established, some startups, lots of new investment. And I think that's exactly what's needed is private to public partnerships and companies to come in to be able to train and hire, and this process will feed back into academia to get more kids who want to do science and technology. And again, the cycle keeps growing. And again, I'm here only 4 days, so I don't feel very comfortable giving advice on how to go forward. But from what I've seen is that there's lots of smart people working at this. At the same time, it does seem like we need much better infrastructure and education, and I know people realize this and are working hard at it. I think one of the keys is, for a small country like us, is to have a clear idea of what we want to focus and specialize in, and become experts at it, and present to the world the ability to educate as well as then harness that intellectual and knowledge to excellence in novel enterprises.
Thank you for the elaboration there, Ardem. And so to help us remember research activity, in terms of the last 30 years, there's many challenges associated with scientific output, but indeed the seeds of increased activity and continuing striking activity are there. Perhaps if you can elaborate the moment of discovery, an example we can take with us here, and involves a story of one of your postdocs in terms of the discovery of piezo. So if you could walk us through this figure here in terms of what that scientific activity looked like on an individual team member level, then we can talk about your laboratory overall as we seek to stand up an increasing number of labs in Armenia. So please, the discovery of piezo.
So I think for me this slide represents one world, and that is resilience. So my postdoctoral fellow Bertrand Coste took on the project of finding these mechanosensors, and without going into technical details, what you're really looking at here is all those black lines are him wasting time on candidates that didn't end up being the right molecule that he was looking for. And to make each of these black bars, he had to spend 2 or 3 days to test something. And the point is that for 11 months, he repeated these experiments again and again on different candidates, and was again and again not successful. But he didn't give up, and kind of my job was to help him both prioritize and stay motivated to keep doing it. And finally, in red, the 72nd attempt at identifying this molecule, he kind of got the right one, and when he got rid of that protein, this activity that we were recording was almost vanished, and so he finally got it. But again, to bring it back, it's a story of sometimes science, you have to persist, right? I don't know if you can imagine this. I mean, I said the words, but if you put yourself in that shoes, where you're working on something every day, full time, and for 11 months not getting any positive feedback from what you're doing, and to still come in the next day and try it one more time. So that's what it took for him, and I think the rewards were fantastic, so I'm glad he didn't give up, and it's an important lesson there.
Indeed, this graph says so much, and in terms of our earlier discussion, the activity of current on the x-axis and the very painful negative results all the way until we have the discovery. You mentioned, Bertrand, specifically, how were you able to recruit such an individual? What support was provided, both as a scientist and a leader? Were grad students attached to him? If you could help us visualize what the build out and support of a lab is, we tremendously appreciate it.
Yeah, that's a very good question, actually, because it's something that in my career it has evolved. And so by the time Bertrand came into my lab, we already had some success from identifying the temperature sensors that we didn't talk much about today. And so in a scientific career, as you become more and more successful, these very young, talented kind of students seek you out. So I didn't have to do anything. He wrote to me and said, "Can I come work in your lab?" And so that was fantastic. But I guess the point of it is that when I first started my lab, students like Bertrand would not come and work for me. And so the work is much, much harder. In those days, I would say, you would hire more diamonds in the rough, and you have to help them become more shiny. And so it's harder in the beginning, and as you succeed more, it kind of gets easier and easier. It's even easier for me now to do good science. So I think maybe the general lesson there is, again, what we talked about, maybe for the whole country, for the scientific community, it is painful in the beginning to start, but once there's an infrastructure, there's a history of success, then it attracts better and better people to come be part of the community.
We'll be opening up for public Q&A in a moment. So if you have a question, please begin to queue at the left or the right microphone. So with this as a follow-on, Artem, you mentioned this aspects of support, the brand of the lab, if I may say, in terms of recruiting talent. Could you talk to us a bit about institutional support? What do universities and institutes need to do to house laboratories?
So in the US, where I am, actually most of professors who do research are mainly in charge of raising funds to do research. And you can do this from private foundations, you can do that from the government. We have a National Institutes of Health, where we write grant proposals, very competitive. The rate of acceptance of a grant is, depending on the year, somewhere between 10 and 20%. So vast majority of grants that you write, spend month writing it, you don't get, so it's very competitive, peer-reviewed system. So there's lots of self-reliance on this aspect. So the government, not the government, the institutions, the universities that we work for also play a very important role. Some give direct support, some support people when their research is good, but for some reason they haven't gotten these competitive grants. And once you become successful, you get recruited from other universities, so your home institution kind of offers you more resources to keep you. So the institutions kind of balance what most professors do, which is raise their own funds from different sources. But I think what the institutions do, which is more important than money, is create the environment for collaboration, for technological advancement. You're really, again, I can emphasize that we don't work in vacuums, and my neighboring professors have a huge influence in my thought process. What questions will we ask next? And these human interactions are extremely important for success.
Thank you, Dr. Patapoutian. We have some questions from the floor. Please.
First of all, let me to welcome to your homeland.
Thank you.
It's our privilege to have you, and it's a lifetime experience to meet the Nobel Laureate, and especially if it is your nation. I have two questions. First of all, maybe it's a very simple question because I'm not a biologist, physicist, but you mentioned that the key element that opens up this feeling of the pain and others is the pressure. When I saw your picture with the rose, for example, my first feeling when I'm watching the rose is the feeling, not by the touching, but in a brain level, feeling the pain that when I can touch it, even without touching, I can feel the pain because it's kind of, I wonder whether the mechanism is the same protein or the same agent are working when I am without touching, can feel the pain. And the second question, in the first part of our meeting, there was a discussion whether the scientists should come to the business with the ideas, or the business should come to the scientists and say, "But I need this." What was because you told us that your discovery can be used for some development of some painkiller. What was the driving force to look for painkillers or the fundamental science?
Sure. I'll start with the first one. So, our brains have evolved to, of course, sense the environment, but our brains have expectations, and this representation of the outside world has clear expectations of what's going to happen, and you use your different senses for this anticipation. And so when you see a rose, you might remember last time you touched a thorn and felt pain, and so you might have a visual of anticipation of that without even activating the sensors. And so this is very common, and it's called the central processing of the peripheral input. So you don't even have to have the input to have the feeling. I think that one very classical example of this is tickling. You can be tickled even if someone doesn't touch you. It's obviously the touching that usually induces it, but if someone just comes close, you might feel that sensation, although there was no pressure applied to your body. So this is very, very common indeed. The second question, I think, was about interplay between basic science, applied, and I think, you know, for example, in the United States, very rich country with, you know, very large budgets for National Science Foundation, NIH, I think there's many, many people who can afford to say, I'm doing basic science and I don't care if there's any application to this. The situation here might be a little bit different, and I think it'd be very wise, given the limited resources, to do the kind of science that there is good application for. There could be budding of biotechnology or tech development, and vice versa. With respect to, again, I'm not a government official. I don't get involved in policy, but I think that this interplay is very important. And I think as long as there's good communication between those who are developing new technologies or just thinking about the basic aspect of this, for those people to constantly talk about and coordinate is a very good thing. For me, personally, I feel like the discovery part, just understanding science for science sake, is the most important thing for me. But I'm also find that the most gratifying thing if my discoveries ever get applied and do get applied for the benefit of humankind, and that's starting to happen, and I can tell you that's the most wonderful feeling. So I love having one foot in both sides, and it's wonderful when it comes together.
Thank you.
Thank you. Alternate mic, please go ahead.
Thank you, Mr. Patapoutian. First of all, congratulations for this prestigious prize, because you know you're the first Armenian who got that prize. We heard a lot of, let's say, theories of conspiracy about the Armenians and Nobel Prize. Thank you for destroying those. And I have not a question, but more requests. So I'm representing some company which is in engineering city in Baghrevand. We have engineering community there. We have engineering school for people and base department for the students. But most important thing, we have there space and science and technology museum, two museums in one place, where we collected all the information about the engineers and scientists. Alexander Kemerjan, his name was called here, with Armenian origin, and a lot of things marked made in Armenia. So we are trying to show to young generation, new generation, everything, the traditions that Armenians put in science and technology. So my request would, I don't know what's your schedule is, if you have a time to visit us, it will be an honor for us. But except that, I would like to ask some small, maybe your picture with your autograph that we could put in that museum, because it's very important for a young generation to see, sorry, but alive, living heroes around them. It's very inspiring, as you mentioned it with the president's example. So that will be, if it's possible to arrange that, we will be very thankful. Thank you.
Sure. I like taking pictures.
And I think your vast Twitter following also likes the pictures you take.
Please email me. I'll be happy to correspond with you.
Thank you very much. Back to the first mic.
Mr. Patapoutian, first of all, thank you very much for being here in Armenia. My company is organizing an international expo in Metaverse. And my question, my company is in business side, but my question is about the science and related to your discovery. Do you think that in the future it will be possible to transfer the senses to the other people who is not just next to you? Just through the metaverse, people will start to feel the touch or maybe the smell or hear. I mean, they are hearing the smell and the taste and the touch. Is it possible in the future?
I think it's possible for sure. There is a lot of research going on in this biological tactile feeling into material science that can give the same sensation. So virtual reality, games to people who've lost limbs, this is something, very active research. And again, one of the ideas is for wearables that gives you the sensation. And so you could imagine very easily that you could hook up someone's experiences into someone else from tactile sensation. Again, this is not my expertise. I'm just learning about this as well, but it's a very active area of research.
Thank you.
Thank you very much. Next question on the mic.
Բարև ձեզ, պարոն Պատապուտյան,
Բարև։
ուրախ ենք, որ հյուրընկալվել եք Հայաստանում։ Հարցը հետևյալն է, ի՞նչն է ձեր գիտությամբ զբաղվելուն ավելի շատ նպաստել, ինչ որ մոտիվացիա եղել է ձեր համար։
I should have worn my translator. I think you asked...
So Dr. Patapoutian, if I may, so what do you find as inspiring? What strength do you draw upon in terms of being able to press forward to do your research to make a difference?
Thank you. I think I really enjoy the process of discovery, which I said multiple times. And I think to me the most important thing is to love what you do. And if you love what you do, then you don't need extra external motivation to do what you do. I've often said that even if I didn't get paid, I would continue to do what I'm doing, which is a luxury that many people can't say, unfortunately, but I encourage all kids to find their passion and do something that they really love, which again is an internal kind of concept of success, that you're doing something that you enjoy. But of course, it would be wonderful if that something that you really enjoy also benefits not just you, but others, the community, and perhaps the whole nation. I hope that answers your question.
Շնորհակալություն։
Շնորհակալություն ձեր հարցը. Next microphone, please go ahead.
Mr. Patapoutian, thank you for being here. I'm so proud of you on the day that I knew that you are one of the laureates of this year's Nobel Prize. I was so excited, and the question that I have since then, that if you could discover the receptors of touch, what do you think? Is it possible to discover the receptors of feeling, longing, or missing?
So what you're talking about is, of course, emotions, and all of that happens in the brain, and it's very complex. And again, it's a very active area of research in neuroscience. The tools in animal models have really, really improved over the last decade, where we can take any neuron or group of neurons and see when they're activated, how are they associated with different emotions, what happens if you block these neurons, does the emotion go away? And it's of course very complex, and the final answer is not going to come in the next year or two, but I believe in the next few decades we are going to understand so much more about the brain and how it experiences emotions, which is of course a very, very interesting question.
Thank you.
All right. Is there another question on the microphone here? Please, go ahead.
Բարոյ Armen Patapoutian, ուրախ ենք ձեզ տեսնել Հայաստանում։ Հարցս հետևյալն է, դուք խոսեցիք կորոնավիրուսի պատվաստանյութի մասին, նույն Մոդեռնայի։ Սովորաբար պատվաստանյութերը միջև մարդկանց ներարկելը փորձարկում են կենդանիների վրա, այնուհետև արդյունքները տեսնելուց հետո նոր ներարկում մարդկանց։ Ինչպես ստացվեց կորոնավիրուսի դեպքում, երբ պատվաստանյութերը շատ արագ կերպով առանց կենդանիների վրա փորձարկելու ներարկեցին մարդկանց։ Շնորհակալություն։
So the question here first is a warm greetings, and the specific example is around coronavirus. The comment is that typically there is a lot of experimentation on animals. How was it possible to be so accelerated during the pandemic times?
So it's a misconception that animal testing was not used for the vaccine development. They were immediately constructed animal models to study just that. And what looked like very accelerated development has actually a history of 10 to 15 years from basic research to development by scientists that were later involved in the Pfizer or the Moderna drug. So what appeared to be very fast turnaround is just actually the fruits, the benefits of the 10, 15 years of research that happened before that. We, in the tech world, sometimes it's called the killer application, and it just so happened that for this technology, mRNA vaccines, the COVID was the killer application that just came, and in that sense, it happened very quickly. But yeah, don't be deceived. It was a decade of research that culminated to that success.
Thank you. We have another question on the microphone here. We'll be wrapping up the open Q&A shortly, so if you do have a question, please queue. We'll be reverting to structured Q&A and closing remarks. And well done, everyone. We are well past the halfway mark. Ardem, fantastic work here. Please, a question.
Hi, Dr. Patapoutian. My name is Tamara. I have a question regarding your research and its applications for pain management. So, given the problem of centrally acting drugs such as opioids, and especially dependency that is usually iatrogenic, and as you're aware, the problem in the US, how do you envision your research being applied in pain management in the future, not necessarily just pharmacogenic, but maybe other applications? Very curious about your vision and how soon you think it'll be translated to practice.
Yes. Indeed, so most pain medications today actually affect the brain, the perception perhaps of pain. And that is exactly why they have side effects like opioids have, they can be addictive and many of the other side effects that they have. So I think the advantage of targeting pain at the gateway, as I've said before, at the periphery, is that if you block these, it will not have any of those addictive side effects that opioid and other medications can have. We have our own challenges. For example, for these mechanosensors, the piezos that we found, they play so many different roles that a general blocker for them might be detrimental. But others, such as one of the previous receptors that we identified, is again very specific to sensing damaging stimuli. And so blocking them might be that sweet spot where you could get the benefits of blocking pain, but will not have other consequences. Again, this is just a hypothesis that's being tested in the clinic now, but as I mentioned before, there are small molecule drugs that inhibit TRPA1, one of the ion channels that we identified, that's being tested in phase 2 clinical trials this year for osteoarthritis as well as intractable back pain.
I look forward to following up on your research. Thank you.
Dr. Patapoutian, I attended your lecture at the Medical University yesterday. I was fascinated with the discovery of piezo 1 and piezo 2, especially for me, who is pretty far from the science, let alone molecular biology. So since today's event is more business-centric or business-oriented, I saved my question for today. I represent the Armenian network state, the pan-Armenian movement. And curious if you have any plans of opening a lab or establishing a lab or research center here in Armenia, and how the network state, the Armenian network state, can help or collaborate with you. Thanks.
Thank you. I don't have any plans right now, but as I said, just visiting here for a week, and it's on day 3, so I have many discussions with both scientists and people in the government, and I'm open to discussions and thoughts of something that makes sense that I can, if I think that my time would be useful and really contribute and it makes practical sense, then I'd be happy to think and consider that, but there's no specific plans as of now.
Appreciate it. If you don't mind, I will email you as well, okay?
Okay.
All right, thank you very much. We'll be closing the open Q&A now, and we're transitioning to our last segment here. And Dr. Patapoutian, as I've been seeing with the Nobel laureates, there are of course laureates in literature, in peace, more recently added economics, physics, chemistry, and of course physiology or medicine. And it seems that once you are a Nobel laureate, you will be asked for your input on anything. And I would imagine that can be anything from food recommendations to life decisions. So I appreciate here we are veering specifically into the economic discussions. So by all means, welcome your engagements, thoughts, and fully appreciate the qualification. Indeed, this visionary session and day hosted by our Ministry of Economy. So as a type of mapping, we discussed different types of scale, meter to nanometer in terms of the activity of piezo. Now, if we consider economic scale and we take your lab and a nation's economy, so for example, Armenia's economy is currently 12 billion, and you've had a previous association with a large firm known as Novartis, revenue at 50 billion. And then you have the operations of your lab, which is on the order of a few million. What insight can you provide in terms of a base level of resource to sustain a research agenda and activity? And what are the implications of those discoveries? If you have a framework of how small elite teams can make a broader difference, and specifically and potentially at national scale.
Again, as you very well said, this is not my expertise, but I think the key is perhaps not the size, but the quality. And although the size matters from the perspective of having enough of a unit, cooperative unit, that they feed on each other and create a product, whether that's a research product or a company product. And you're right, the economics of this is very important, and I'm not going to get into the dollars and cents, but I think if I was making these decisions, I would look at our budget and say what areas of science and technology that we have the bandwidth to become a leader in the world. Because I don't think it makes too much sense to invest a little bit in an area that we know needs 1,000 times more investment to make a dent in advancing that scientific or technological question. I think that's all I really have to say, other than being very strategic and focusing on areas that we can be excellent on. I think I can't emphasize that enough.
So we're seeing here in Armenia, and Garik made the argument earlier about space and tech. So there's a few intersections, and if I may draw analogy to biotech, probably heavy manufacturing of rockets, not an immediate priority, but computer vision in aerial imagery probably makes a lot of sense. Similarly for biotech overall and supporting areas such as bioinformatics, if you could even break apart some of the various research activities you've done, which might be ones a country such as Armenia should focus on sooner rather than later?
Yeah, again, I'll focus on my area of expertise, which is biomedical research. And one of the reasons that, so someone working on tech, for example, IT, what do they need? They need their brain and they need a powerful computer usually. And those are, dollar value, not very expensive. But to do the kind of research that we do, you need microscopes that are multi-million dollars sometimes. You need all this infrastructure that costs a lot. And one of the other problems of biotech challenges is that the arrow between idea and a product is not easy, not short, often requires very expensive clinical trials. And so whether you like it or not, the economics of a question needs to be addressed. And that's why you mentioned this, I'm in the health sciences arena. I think a great opportunity is this idea of bioinformatics or data sciences where, because in biology we're now sequencing the whole human genome, sequencing people who are afflicted with diseases and identifying what small changes in your DNA causes this. All of this creates massive amount of computer data that need to be analyzed correctly, and there's so much information to come out of that. And this is something that, again, dollars and cents is not as expensive as some of the other research to cure diseases, and something that I think Armenia can play a major role. But again, this is just my initial opinions from what I've been looking at, and many more people who are very well qualified are looking into these questions.
So bringing it to the laboratory activity of grant writing as an example, in Armenia's history, there's been strong tie-ins in terms of broader technological infrastructure. So the memory of science is there, but even in the earlier era, grant writing was not there at scale. What specific insight can you provide? And if you could tie it to leadership and the learning activity that grant writing represents for a lab head to support a lab, and how to think about this.
Yeah. So scientists, at least in science that I grew up in, has this huge devotion to anonymous peer review. And what I mean by that is that, again, when we write one of these grant proposals, or if you write a manuscript that you want to be published in a high-impact journal, it goes out to other scientists who review it, and almost all the time you don't know who they are. And these are on a volunteer basis, so we kind of review and critique each other. And this is thought to be one of the most important aspects of coming up with the best ideas of where to invest, what to publish, reviewed by your peers anonymously. And again, I can't emphasize how important of a process this is in science to make sure there's no preferential treatments, there's no money given to friends and family. Instead, it's done on merit and merit only. Some are even experimenting with complete blind review process, where if I submit a grant proposal or a paper to be published, they might not show my name to the reviewers. So if they like me or dislike me, they won't have an effect on the process. So I think when we talk about the scientific method, impartiality and looking at it for its own value is so important, and creating this environment where science is being looked at just for its merits is absolutely essential for the process to work well.
Given these critical activities, peer review, developing a proposal, what recommendations would you have to budding scientists or scientists that are looking to return to scientific activity? Is it best to find a mentor? Are there stepping stone steps? What would you recommend if someone has the aspiration to lead a laboratory?
Yeah, one can never underestimate the importance of having mentors. When I think back about my career in the beginning, and I was an undergrad technician working in a laboratory, a couple of PhD students took me under their wing and pretty much taught me how things work, how science works, and I have so much gratitude to them. So it doesn't necessarily have to be a famous professor that's your mentor, but there's lots of important people along the way in your career. So finding a good mentor who would advise you correctly is, of course, very, very important. The other thing you talked about, grant proposals, I think it's just so important to have many ideas and try them all, because some people get very fixated on the first few ideas that they have. And one of the things that science teaches us is that most of your great ideas are not going to work. And so you have to be very flexible and keep thinking of reinventing yourself, thinking of different ways to approach the same question. I found it very important in my career.
And from your humble beginnings as a tech to a Nobel Laureate visiting us today, how do you think about the life cycle of a scientist? Presumably folks you have trained have gone into industry or done things outside of science. Sometimes they may come back. Do you have any thinking around the workforce aspect of this, either on an individual story level or a model in how a country can think about its scientific capacity in terms of its people?
Can you repeat the question again? Sorry,
Sure.
didn't quite get it.
So, for example, there's the lab tech, grad student, the postdoc, the double postdoc, the assistant associate full professor, and presumably this is an upgrade path, but people can veer off, do something else.
Yes.
How do you think about this, and how do you think about it in terms of the workforce overall, collections of labs, how that supports the economy? So two scales of labor.
I mean, in biological sciences, when I first started, there was this academia work was put on a pedestal where that was the main goal, and people who went into translational or industry work were kind of, they did not follow the path, right? I think that's pretty much wiped out now. I think nobody thinks that way. And all aspects of this is interconnected and important. If we just did basic science and nobody translated it, it would be very much big waste of time and energy and resources. And so I think, with respect to the different aspects of careers you talked about, they all play very important roles. Scientists get trained in a laboratory usually, not in a classroom. And so these stages are very important to experience doing science in real life, because you're probably not going to learn it from a textbook. And again, they all play a very important role in the progress of science.
Earlier today, Marilu Papazian mentioned education. And what are your thoughts, considering well before the university level, perhaps drawing from your own experience in Lebanon originally and subsequently at different post-secondary institutions? How do you think about education, and what recommendations would you have in terms of systemic education to support science?
I think we touched upon this. Education is the most important framework for a society, I think, and I'm talking about from kindergarten to high school to activities like places like TUMO does here, and all the way into college. I think early on, just a great general critical education is best, but again, for a place like Armenia, for postgraduate, some specialization and critical focus is of great use.
Dr. Patapoutian, we have had quite the journey here together, starting from Lebanon, going through a crash course in ion channels, to the implications of biotech and tech in the economy. We'll invite you, if you have any closing remarks, as we look to round out this session here. And again, it's been an absolute privilege. Please, any last comments for our audience?
I think we covered everything. You're probably bored the audience a little bit, and it's been a long day for you, but I want to thank you for this very important questions that you asked, and just want to thank everyone here from the Minister of Economy for inviting me, for all of you for being such generous hosts, and again, what a privilege for me to come and experience this, and thank you for staying and listening and participating. Thank you.
Thank you very much.
Շնորհակալություն հարգելի բանախոսներ։ Գիտության եւ գործարարության օրեր 2022 միջոցառումն ավարտվում է եզրափակի Հայաստանի Հանրապետության էկոնոմիկայի նախարար Արմեն Քերլոպյանը։
Շատ տպավորիչ էր։ Ես ցնցված եմ ուղղակի մինչև այսօրվա խոսակցությունը դժվար է պատկերացնել ձեր արած հայտնագործության մասշտաբը եւ էությունը ու այս առումով իսկապես շատ շնորհակալ եմ որ ժամանակ գտաք Արտեմ ջան եւ այցելեցիք Հայաստան, մասնակցեցիք բազմաթիվ միջոցառումների եւ դեռ պիտի մասնակցեք եւ ձեր խոսքի մեջ ինձ թվում է դուք ասիք այն արտահայտությունը, որը իսկապես կարևոր է եւ ասիք, որ միգուցե կառավարությունը ձեզ հրավիրելով փորձել է ցույց տալ, թե ինչն է կարևոր մեր համար եւ իրոք ես համաձայն եմ ու կրկնում եմ ձեզ, մեր համար իսկապես ամենակարևոր բանը ցույց տալ, որ գիտությունը շատ կարևոր է մեր համար եւ ձեր օրինակով ցույց տալ երիտասարդներին, թե ինչ ճանապարհ է նախընտրում կառավարությունը, ինչ ճանապարհ է նախընտրում այսօրվա Հայաստանի հասարակությունը։ Եվ կարծում եմ, որ ձեր այս մի քանի օրվա Հայաստանում տարբեր միջոցառումների մասնակցելը իսկապես բացում է մեր հասարակության մեջ այն բջիջները, որի մեջից պետք է նոր գիտնականներ ծնվեն, որի մեջից պետք է նոր գիտական հայտնագործություններ ծնվեն, որը թույլ կտա, որ մեր երկիրը, մեր երկրի տնտեսությունը իսկապես բեկումնային զարգացում ունենա։ Ես եւս մի անգամ շնորհակալություն եմ հայտնում ձեզ, շնորհակալություն եմ հայտնում պրոֆեսոր Արմեն Քերլոպյանին հրաշալի մոդերացիայի համար, իսկապես շատ տպավորիչ էր։ Շնորհակալություն եմ հայտնում այսօրվա մյուս բանախոսներին, Ardem Patapoutian, իր թվային տնտեսության վերաբերյալ հրաշալի զեկույցի համար, Ardem Patapoutianին, Գարիկ Իսրայելյանին, առաջի պանելային դիսկուսիայի բոլոր մասնակիցներին, կարծում եմ, որ շատ հետաքրքիր օր ունեցանք եւ այսօրը մեզ շատ նյութ տվեց մտածելու, վերաիմաստավորելու գիտության դերը մեր երկրի հասարակության պատմության մեջ, երկրի տնտեսության մեջ, կարծում եմ, որ այս տիպի քննարկումները թույլ են տալու, որպեսզի մեր երիտասարդները տեսնեն ընտրություն, գիտությունը որպես հնարավոր ամենալավ ընտրությունը։ Շնորհակալություն եմ հայտնում ձեզ, հարգելի ունկնդիրներ, հարգելի մասնակիցներ, մի քիչ մեր հասարակությունը սովոր չի, ամբողջ օրը գա այսպիսի միջոցառումների, չարաշահել ենք երևի համբերությունը, բայց կարծում եմ, որ այս կոնֆերանսները, այս համաժողովները պետք է դառնան օրինաչափություն եւ մարդիկ գան, տրամադրեն ամբողջ օր լսելու, սովորելու, իրար հետ շփվելու եւ իհարկե շատ շնորհակալություն եմ հայտնում նորից բոլորին, մաղթում եմ ամենալավն ու բարին, այսօրվա այս միջոցառումը հայտարարենք փակված եւ իհարկե մենք ունենք ուրիշ միջոցառում այսօր իրար հետ գնալու, շատ շնորհակալություն։